All right, welcome to The Explainer. Today, we're diving into a high-stakes drama that's happening right now inside basically every one of us. It's this constant, microscopic battle for control, a genetic tug-of-war that really determines our health. So, yeah, let's get into it.
So, think about it for a second. You are made of trillions of cells, and they're all supposed to be working together in this perfect harmony. What is it then that stops just one of those cells from going rogue, from just completely ignoring the rules and trying to conquer the whole system? Well, the answer is inside this incredible, but as we'll see, sometimes fragile set of genetic controls. You see, our bodies are built on a really simple but profound principle: controlled growth. I mean, from the moment we start as a single cell, our genes are giving out these super precise orders: when to divide, when to grow, and, maybe most importantly, when to stop. It's like this beautifully orchestrated symphony happening inside of us all the time. But what happens when that symphony gets disrupted? You know, when the sheet music gets all scrambled? That's where our story really begins. Because at its very, very core, cancer is a disease of broken rules. It kicks off the moment the genetic code that keeps our cells in line takes on some damage. Now, to really get this, we need to meet the key players in this drama. And first up are the genes that are actually supposed to encourage growth. In a healthy body, they're absolutely essential. But when they get mutated, they become the villains of our story. So here we have two sides of the same genetic coin. On the one hand, you've got proto-oncogenes. The best way to think about these is like the accelerator pedal in your car. They help the cell grow and divide exactly when it's supposed to. But when a mutation hits, they can flip and turn into what we call oncogenes. And now all of a sudden, that accelerator is stuck to the floor. The cell is just getting this constant, relentless signal to divide, 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 with no off switch. But don't worry, our bodies aren't defenseless. We have this amazing defense system, a whole set of heroic genes designed specifically to slam on the brakes when things start to get out of control. So let's meet the body's genetic guardians. These are the tumor suppressor genes. And their entire job, their whole reason for being, is to act as the body's internal police force. They're constantly patrolling our genome looking for any sign of trouble, and they're ready to shut down any cell that even thinks about becoming cancerous. So what do these guardians actually do? Well, you can boil it down to three core missions. First, they can tell a cell, hey, slow down, stop dividing. Second, if they find some damage in the cell's DNA, they can pause everything and call in the repair crews. And third, if the damage is just too severe, too far gone to be fixed, they have one final drastic option: ordering the cell to self-destruct for the good of the entire body. Okay, so we've got genes that push the accelerator and other genes that slam on the brakes. It's a really elegant system. Cancer happens when this system breaks down, specifically when the guardians fail. Let's take a look at exactly how that can happen. Our first guardian is a gene called RB, and you can think of it like a security guard standing at a really critical gate, the checkpoint where a cell has to commit to copying its DNA and dividing. The RB protein physically stands in the way, blocking another factor called E2F from opening that gate. But if the RB gene gets mutated, the guard is gone. The gate is just left wide open, and cells can rush right through to divide whenever they want, completely without permission. Now, this next one is really interesting. This is arguably the most important guardian of them all, the P53 gene. It's often called the guardian of the genome, and for good reason. When P53 senses DNA damage, its first move is to hit the brakes on the cell cycle, which gives the cell time to make repairs. But if the damage can't be fixed, P53 has to make a really tough call. And that final command is to initiate something called apoptosis. This is the official term for programmed cell death. And it's not some messy, chaotic explosion. It's a very clean, very orderly self-destruction sequence. P53 basically tells the damaged cell, look, you are a danger to the whole organism. It's time to sacrifice yourself for the greater good. So the most crucial point here is what happens when this ultimate guardian fails. If the P53 gene itself is mutated, it goes blind. It can't sense the damage anymore, and it can't issue that self-destruct order. And now, the damaged cell is totally free to just keep dividing, passing on all of its errors and mutations to every new cell. It becomes, for all practical purposes, immortal, a rogue agent that has lost its most fundamental safety switch. And the failure of this one gene is devastatingly common. Just how common are we talking? Well, take a look at this number. 50%. 50%. That's because, as the data shows, the p53 gene is the single most frequently mutated gene in human cancers. A broken p53 guardian contributes to half of all cancers. I mean, that one fact right there just shows you how absolutely critical this single gene is to protecting us every single day. So this entire battle is raging on a hidden genetic level. But how do we actually see the evidence? How can we find the fingerprints that this cellular crime leaves behind? Well, this is where modern medicine gets to play detective. We do it using something called a tumor marker. Now, these are basically substances, usually proteins, that are either produced by the cancer cells themselves or by our own body in response to the cancer. You can think of them as forensic evidence, the clues left at the crime scene that tell us a battle has been fought and that the rules have been broken.
And these clues are incredibly useful in the real world. I mean, doctors use them to help diagnose a cancer, of course, but they also use them to figure out a prognosis or to monitor treatment. For example, if a marker level isn't dropping, that might be a sign to try a different therapy. They're also used to check if a cancer has come back, kind of like an early warning system, and even to screen people at high risk before they even have any symptoms. Let's just look at a few quick examples to make this real. You've probably heard of PSA. That's an enzyme used as a marker for prostate cancer. Or how about BRCA1? That's actually a mutated tumor suppressor gene itself, and finding it is a huge red flag for a high risk of breast and ovarian cancer. These markers create a really tangible link between the genetic chaos happening inside a cell and a real diagnosis in the clinic. So from stuck accelerators to failed brakes and broken guardians, we can now actually read the genetic story of how a normal cell turns into a cancerous one. We can find its fingerprints in the blood. The science has brought us this far, which leaves us with just one final provocative thought. Now that we can read cancer's story, what are we going to write in the next chapter?